Hi, everybody, and welcome to what day is it? Thursday today. We are going to learn a little bit about blood types, and we're going to use Punnett squares to figure out your blood type. How many of you even know your blood type? Next time you go to the doctor, ask them because that's actually an important piece of information to know. Guess what I am? I am an A plus, A positive. Anyway, okay, so blood types. Let's talk about that. There are four blood types. There's type A blood, type B blood, and or type AB blood, and then type O. Blood types A and B, they are dominant. So the A and B, they are dominant over O. The O is recessive. And when you have A and B together, they are both dominant together. One isn't dominant over the other. So we call that codominance. So that's how you can get the type AB blood. So how do we write these blood types when we're doing Punnett squares? So there's two possibilities for type A blood. You can see right here, big A, or big A, little I. That little I is going to represent type O blood. If you have little I, if that was your genotype, that would mean your phenotype then would be type O blood. So you can see in this genotype right here, both capital letters. So that would be homozygous dominant for type A blood. And then right here, we would say heterozygous for type A blood. Heterozygous, and it's type A because A is dominant over the O, and heterozygous because it's two different letters. Why do they use an I for type O blood? I don't know. They just do. Okay, so type O blood is little I, and that's recessive. How would you write homozygous for type A blood? So if a person was homozygous for type A blood, how would you write the genotype for that? If you look up here, it's one of these two possibilities. Which one do you think it is? 50-50 shot. Right there, that would be the homozygous for type A blood. Okay. Now, okay, yeah, there you go. Oh, Mr. Nip gets to make an appearance in there. And then so there's also something else with your blood types that we're really not going to deal with. But if you notice in the last slide, I just talked about type A blood, type B blood, A, B, or O. I didn't talk about the positive or negative part of it. It's called an Rh factor. And um, what what that means is if you have a positive, you have this coating around your blood cells. It's like this protein coating that's around it, and if you have it, then you're a positive. You test positive for it. If you don't have it, then you test negative for it. When we do our Punnett squares, we're not going to worry about the positives and negatives because it just makes things even more confusing. This doesn't have any other examples on here because it says next to go take the formative. But I just want to go over a few other little things. So right here it has the two ways to write type A blood. What are the two ways to write type B blood? Ooh. These are the two possible genotypes for type B blood. So this would be the homozygous, big B, and this would be heterozygous. Sorry, there's a glare there. Big B, little I. Heterozygous, but it's type B because B is dominant over that O recessive allele. And so that's what that would be. What are the possible genotypes for type AB blood? It's just that that's the only possible like genotype you could have for type AB blood. Remember, they're co-dominant together. A isn't dominant over B. B isn't dominant over A. They're dominant together. And then what's the last one? Oh, what are the possible ways to write type O blood? Remember, type O blood is recessive, so the only possible genotype you could have for that is little I, little I. Okay. So that is that, I think. Hopefully that made sense. If you got questions, um, respond in the Flipgrid, and good luck on the formative. Hope y'all are doing good. Bye. Oh, this always takes me a minute. And okay, bye.